Hi everyone, and welcome to this lecture and welcome to this chapter where we are going to talk about the clonal selection algorithm. And what we have to say is that the clonal selection algorithm belongs to a general class of algorithms inspired in the functioning of the immune system, which is artificial immune system. So this is a class of algorithms inspired by the vertebrate immune system. We have four algorithms that are most discussed in this class, in this general class of algorithms. We have the clonal selection algorithm, which we will discuss here and apply a project. Negative selection algorithm, algorithms based on immune cell networks, and algorithms based on the function of dendritic cells. The two first algorithms, the clonal selection algorithm and the negative selection algorithm, are the most frequent ones found in applications. This algorithm resembles the genetic algorithms in many aspects. So this is similar to the process of Darwinian evolution that occurs with populations of individuals, but in this case it's population of antibodies. In this case we have the maturation of antibody and T-cell receptor response to foreign antigens. So this is the process in which this algorithm is inspired. So here we have combined and successive processes of hypermutation and selection of antibodies in their respective cells refine the recognition by the antigen. The main applications are, first one, classification, because the immune system's main task is to recognize agents and the self-antigens in our body. It can be used for tasks of classification. It can also be used for pattern detection, anomaly detection, and information security or cybersecurity. And we can also use this algorithm for optimization of mathematical functions, just like most other algorithms. So here we have the biological basis of the clonal selection algorithm. In our bodies, the B lymphocytes are responsible to produce diverse antibodies. So we have a lot of lymphocytes, and each one of them will produce a different antibody. And here we have an antigen. We have different affinities with these antibodies. Let's suppose that for this antibody right here, the affinity is the best one. So this antibody and the cell that produced this antibody will be stimulated to be cloned. So here we have memory cells, and here we have the plasmocytes, which are the cells that produce these types of antibodies. So the cells that produce the antibodies with most affinity are stimulated to reproduce more. The cloning of the antibodies in the respective cells is proportional to the affinity. So let's suppose that these are the two best antibodies. The affinity of the green antibody is 60%, so it will produce an X number of antibodies. The affinity of this antibody is 80%, so it will produce even more antibody than this one. And the worst antibodies will produce just a little amount of clones. So let's suppose that these are the affinities of the antibodies. The sum is 335. If we divide these numbers by the sum, we have these ratios or these proportions. Let's suppose that the antibody population is 50. So this is the number of clones that each antibody will have. You can realize that for this violet antibody that has the best affinity, we produce more clones than the other antibodies. We will see this calculation with more detail in a further lesson. And we also have the hypermutation of the antibody. The antibodies will be stimulated to reproduce, to be cloned, or their cells. And here we also have variations in these antibodies. However, this variation is inversely proportional to the affinity of the antibodies. The better the affinity, the lower the degree of hypermutation. So worse antibodies will suffer more mutations than best antibodies. This was a general overview of the biological basis behind the clonal selection algorithm. In the next lectures, we will see with more details the steps of this algorithm. I see you in the next lecture.